Hello, everyone, and compliments of the new year. Thank you for joining us, and welcome to the first edition of the GSU Malaria Journal Club monthly series for 2025. Today's webinar is a vector-focused one, referencing Marilou Boddé's recent preprint titled Genomic Diversity of the African Malaria Vector Anopheles funestus, as you can see on the screen. My name is Paballo Chauke, and I'm a training coordinator at Malaria Gen, working with a team of great people to organize these monthly events, and we are excited for the 2025 edition. So thank you so much for joining us from wherever you are in the world. Just a few ground rules. The Journal Club is being recorded and will be available on our YouTube channel later. So please do check out the Malaria Gen YouTube channel, and you can share the recording with your friends, your networks and colleagues, or you can rewatch for yourself. Please kindly stay on mute so as to not disturb our speaker. We will have a Q&A session at the end, as well as discussion where you can raise your hand or type your question in the question and comments or the chat box as well. Without further ado, I would like to introduce our speaker for today, Dr. Marilou Boddé, who's currently in Germany, but she works also in Madagascar and used to work at the Wellcome Sanger Institute. So we know her quite closely, and she was a TA for our vector training course. And I'm glad that she enthusiastically agreed to give the first Journal Club for 2025 when I invited her. So thank you so much, Dr. Marilou. The title of the talk is, as you can see on the screen, Genomic Diversity of the African Malaria Vector Anopheles funestus, similar to the preprint. I'm kindly going to read a short bio for Dr. Marilou Boddé before she begins her presentation. Dr. Marilou Boddé is interested in developing and applying computational methods to monitor wild mosquito populations through genome sequencing data. She led data analysis for the project investigating the genomic diversity and population structure of Anopheles funestus throughout much of its species range, using the whole genome data. She also developed species assignment methods for ANOSP, a targeted sequencing approach to identify any species in the Anopheles genus. Currently, she's working to combine ANOSP sequencing data of tens of thousands of mosquitoes with associated metadata to investigate signals beyond species assignments and identify the benefits of incorporating sequencing into vector surveillance. Dr. Boddé is involved in the early stages of planning a landscape genomics project studying the relationship between genomic structure and the environment across multiple species of Anopheles mosquitoes. Dr. Marilou is a postdoctoral fellow at the LIB in Bonn, Germany, and the IPM in Antananarivo, Madagascar. I'm currently advised by Dr. Diego Ayala, as well as Dr. Mara Lonichak. Marilou, thank you so much for joining us. You may begin your talk.
Thank you very much, Paballo. Thank you very much for this very kind introduction. So I'm Marilou, and today I will talk about our preprint genomic diversity of the African malaria vector Anopheles funestus. The preprint is online since last month, and it really has been a very exciting collaboration between a lot of people. So here in tiny print, you can see all the people who contributed to this. And I'm very pleased to be presenting that on behalf of all of them for you today. All right, so let me see. Okay. Yes, so Anopheles funestus is one of the major malaria vectors in sub-Saharan Africa. It's one of the 4 major malaria vectors. The other 3 are Anopheles calitziae, Anopheles gambiae, and Anopheles arabiensis, which are all part of the gambiae complex. And Anopheles funestus and the gambiae complex diverged approximately 30 million years ago. And the gambiae complex we all know and love because about 10 years ago, since in the last 10 years, there has been a huge effort to try and generate genomic resources for the gambiae complex with reference genomes and mostly a lot of short-read whole genome sequencing. And through the generation of these resources, we've learned so much about the gambiae complex, from its population structure and the amount of genetic diversity they have, to new and updated information on species ranges, cryptic species, as well as the emergence and spread of insecticide resistance, and the identification of potential targets for gene drive. Up to now, such an elaborate, genomic understanding of Anopheles funestus was lacking, and that's the gap we're trying to fill. So why is it important to also generate these genomic resources for Anopheles funestus? Well, because like the gambiae complex, it's a very important malaria vector. It has a large geographic range, as you can see here on the right, that also covers many different habitats, so it has quite an adaptive potential. But apart from that, Anopheles funestus is really an excellent malaria vector. It's highly anthropophilic. It has very high plasmodium rates. In some places, it has an extended transmission season compared to the gambiae complex. It also has a slightly longer lifespan than the members of the gambiae complex. And in much of southern and eastern Africa, it's now the dominant vector. And in reaction to indoor-based malaria control interventions, it's shifting from nighttime indoor biting to either outdoor biting or earlier in the evening or later in the morning biting. So these are types of behavioral resistance, but it's also developing genetic insecticide resistance, which we will come to talk about. Okay, so what have we done? We have sequenced 656 mosquitoes that passed all quality controls, and they have been collected from 13 different countries in sub-Saharan Africa, and they span a large amount of the geographic range of Anopheles funestus. So here on the left, you can see the approximate range of funestus in gray, and then in all these different colors, we have the collection locations from which we have mosquitoes that we sequenced. So with this data, we did to see what is the major structure within the data set, we did a principal component analysis, PCA, as you can see here. And from this, we see that the strongest signal, so principal component 1, splits the data basically in two groups of samples, where we have sort of samples from around the equator here, over here on the left, and then we have samples from south of the equator that form a couple of different smaller subclusters. Another thing that you really notice is that the second principal component basically pulls out only one geographic cohort, namely the samples from North Ghana. And if we now turn our attention over here to figure B, this is for each individual, we have looked how many heterozygous sites does the individual have, and they have been ordered by population. And that is a measure for the amount of genetic variation that you find in each of the populations from which these mosquitoes come. And you can see sort of the same, similar pattern where we have a bit of a split between the samples from the collected from around the equator, which have slightly higher number of heterozygous sites, so a bit more genetic variation, compared to those samples from Central, Southeastern Africa and South Mozambique. But what you also see on this part is that samples from North Ghana really have a big decrease in the number of heterozygous sites and the amount of genetic variation that they have. And we also, I don't show it here, but we look at runs of homozygosity, which is another indicator for how much variation do you find in one population. And it has very long stretches of homozygous parts of the genome within individuals. So what we think happens is that there has been a severe bottleneck in North Ghana, and that is really shaping the genetic structure that we see there. Okay, so if we turn our attention back to this PCA, we see that there's a split between samples from the equator and samples from the south, and North Ghana kind of stands out. But what's quite striking is that these equatorial samples from South Ghana over here in the west to these samples from around Lake Victoria pretty much sit on top of each other in the PCA. They really form one tight cluster, meaning that they have a lot of shared variation, which could be indicating that this really acts as one mixing population, connected and mixing population. So if you do a PCA just on the samples collected from around the equator, you can see a bit better that there is some geographic structure. So over here we have samples from around Lake Victoria, and then over here we have in green South Ghana, and in brown Nigeria, and in red Cameroon. So there's a little bit of geographic structure, but really it seems to be a very well-connected population. Okay, so this PCA was done on one chromosome arm, chromosome arm 2L, which does not have any of the common well-known inversions, and therefore serves as a good region to sort of study the background or neutral structure along the genome. But what we're now interested in is to see how does structure vary along the genome and which regions of the genome are potentially interesting. So for that, we did a sliding window PCA. So for sliding window PCA, we basically chop up the genome into little windows. And in each window, you perform a PCA. But contrary to the PCA I just showed you, we only care about the position along the first principal component, and we forget about all the higher components. And then you can plot that like this. So along the x-axis, we have the genomic position in a base pair. So that's basically the center of each of these windows, if you will. And then on the y-axis, there is the PC1 value for each individual. And we connect, for each individual, we connect its PC1 values throughout all the windows. So each line corresponds to a single individual. And here we've colored the lines by the geographic cohorts, so by the collection locations that were shown on the previous slide. Yeah. So each line represents one individual, and now we can look along the genome, and in each little piece of the genome, we can see what does the structure, what does the driving, the most important structure look like that in this piece of the genome. So before I showed you a PCA on chromosome arm 2L, which does not have any segregating inversions, so it's kind of a boring arm or good background structure. And if we look on most of these arms, in most of this arm, we see roughly two bands of lines. So lines are individuals and bands are then groups of individuals. So on the bottom here, this is the line that corresponds to the equatorial cohort. Here on the top are the more southern cohorts, and you can even see a little bit this substructure where we have the central cohorts with Gabon and DRC, Kinshasa, and then on the top we have the southeastern cohorts. We can already see that on this boring arm there is actually something going on already. And this peak corresponds to a selection site where an insecticide resistance allele has been selected, but we'll come to talk about that later. Okay, so then let's look at another arm, for instance, chromosome arm 3. Here we do have two segregating inversions, and you can see that along the sliding window PCA that that in these inversion regions, the structure looks very different from this background structure where you have the equatorial cohorts versus the southern cohorts. One thing I didn't mention is that the samples from North Ghana that went through this bottleneck are not included in this PCA because they add a lot of noise to what we see, so they have been excluded. Okay, so let's take a closer look at the inversions. So here I'm showing a plot that is computed on the same data as before, but I've selected just two individuals from two cohorts so that it's a bit more visible what we're talking about. But it's the same data and it's been computed on all samples. Okay, so what is an inversion? An inversion happens when if you have a chunk of the genome and that kind of flips and reinserts back into the genome. So what then happens if you have an individual that is heterozygous for the inversion, so it has one chromosome that has the ancestral orientation and one chromosome that has this flipped orientation, then if a crossover event happens within this inversion region, then if this recombination happens, then the gametes that come from this recombination event have either 0 or 2 centromeres and are therefore not viable. So in practice, that means that recombination is really suppressed between the ancestral orientation and this flipped inversion orientation. And then over time, both of these orientations will aggregate variation independently and sort of set off on separate evolutionary trajectories. And that can be really important, for instance, in speciation or adaptation. But what it means in the context of, we can also really see this in the context of PCA, because each of these orientations will have aggregated variation over time, independently, differently. So with this PCA, we're looking at what is the driving structure within each region of the genome. This inversion karyotype is really important for the structure. So if we look at, so here I have the same data as the previous slide, but only on two cohorts, Malawi in blue and Cameroon in red. And if you look outside of the inversion region, you can see the same background structure that we talked about before, where Cameroon is in the equatorial cohort, Malawi is in the southeastern cohort, and they sort of form these two bands of samples. But once we go into the inversion region, if we restrict our attention to Cameroon in red, you can see these clear three different trajectories, and they correspond to the three possible inversion karyotypes of the individuals. So in this case, we have a bunch of individuals that are homozygous standard for this 3RA inversion, a bunch that are heterozygous, and then on the bottom we have those that are homozygous inverted. And the same thing happens in blue. So again, we have the samples, they split into three different trajectories corresponding to the three inversion karyotypes for this inversion. However, you might notice that even though they both have three trajectories, that they don't exactly overlap. So the heterozygous from Malawi seems to overlap with the homozygous standard from Cameroon. And that's mostly an effect because we are trying to squeeze a lot of information into one dimension, essentially. So here is a PCA that is just, that's a 2D PCA that's done on a little part, a little slice of the genome over here. And then you can see that in this window, we really capture both of these phenomena. On one hand, we capture the inversion karyotype that are here indicated by shapes. So we sort of have these three stripes. But on the other hand, we also capture the background structure that we've talked about before, where Cameroon and Malawi are in different groups. So because it's a compound signal, it looks like Malawi heterozygous and Cameroon homozygous standard overlap, but that is just an artifact of trying to squeeze all this information into one dimension. So let's look at another inversion. There's another inversion on chromosome arm 3R, namely 3RB. And again, this is data that has, the sliding window PCA has been computed on all samples except those from northern Ghana, but I'm showing you only 2 cohorts in this case. It's Gabon in orange and Mozambique Maputo in turquoise, teal, light blue. And what you might notice in here is that there's a couple of lines that start out in one trajectory, for instance, here in the middle, so heterozygous trajectory. And then for a small amount of the inversion region, they change to a different trajectory. So this is the trajectory that corresponds to being a homozygous standard for the 3RP inversion. And I just told you that recombination events will not produce viable gametes if the recombination happens between the ancestral orientation and the inverted orientation. But if you have a double recombination event, so if recombination happens twice within the inversion region, you can produce viable gametes. And in that way, the orientations can, the inversion orientations can exchange a little bit of the variants that they have. And it's quite interesting that here we seem to be able to really identify these individuals that either they themselves or recently underwent, had a double recombination event. Okay, so to check that this is really what's happening, here we do another PCA that's done in 1 megabase pair that's sort of centered on the peak here of this one sample that's changing from the heterozygous to the homozygous band. And the samples here are colored by their inversion karyotype. That's on the whole inversion. And you can see here that there is one sample that is heterozygous overall, but on this little part of the genome, it clusters with a homozygous standard cluster. So that's one of these double recombinants. Over here is quite interesting because on one end, we have one of these double recombinants from Mozambique Maputo that joins from the homozygous standard to the heterozygous band. So that's this purple sample that goes from purple to the green cluster. But we see at the same time that in Gabon, sort of all the samples that are homozygous standard or heterozygous seem to be moving to look like homozygous inverted. And that might be the case that there is selection acting on the variation that is found within an inversion. And that has been shown before, for instance, in Anopheles gambiae, there is an insecticide resistance gene that's found on one orientation of the 3LA inversion, and that has probably through double recombination been able to insert itself in the other orientation. So you get selection for, in this case in Gabon, it looks like there is some selective pressure for individuals to, at least in this small part of the inversion, look like they are homozygous inverted. Okay, so with the sliding window PCA, or really also with normal PCAs, you can karyotype all the individuals for the inversions that we find. And in this data set, we find 6 segregating inversions, and we've karyotyped all the individuals. And one thing you might notice from here is that even though there is not very strong geographical patterns, there is one cohort that sort of jumps out in its karyotype, and that is the cohort from Benin. Because first of all, it often has quite a different dominant karyotype from its neighbors. But also for these 3 inversions, it's actually fixed in the standard arrangement. And that is something that has been observed before, namely in Burkina Faso. In Burkina Faso, there are 2 ecotypes of Anopheles funestus. One of them looks to be like the normal continental funestus that we find in many other places, but the other one can particularly stands out in its inversion karyotypes because it's fixed for the same fixed standard for these same 3 inversions. So we wondered, because Benin is not too far away from Burkina Faso, whether we might have found the same karyotype in Benin. But as has been shown from work from Scott Small that was published, I think, 2 years ago, where they did genomic characterization of the Carabina and the Falonzo, the other ecotype in Burkina Faso. The differentiation between these ecotypes is not limited to the inversions alone. They really see a clear signal of differentiation of the ecotypes throughout the genome. So what we did here was to do another PCA on the neutral, non-inversion containing arm 2L to see whether our Benin samples look like the Carabina samples from Burkina Faso. So we took the samples that were published by Scott Small in 2021 and aligned them to a reference genome and called variants in the same way. And then we did a principal component analysis with the equatorial samples, as well as our Benin and North Ghana samples, and those samples from Burkina Faso. So what you can see here is that the first principal component really splits out North Ghana, the second really splits out Benin, and Carabina and Falonzo actually both cluster with the equatorial cohort. Now, once you do a PCA on only the equatorial cohort, then it's clear that the Carabina samples are distinct from Falonzo and the other equatorials. But this is a very clear signal that our samples from South Benin are not the same as the Carabina samples. South Benin also has a very strong peak of differentiation on part of the genome. So around here, the samples from Benin are really very different from all other samples. And we've looked, we couldn't really find any signals of selection there. So it could be some very local adaptation. It could be introgression. We don't really know what's going on, but it sort of adds to the case that we want to make that South Benin is really a very different population from all the other Anopheles funestus that we have in this data set, and particularly very different from its neighbors, and potentially could represent a new ecotype. Okay, so now let's turn our attention to signals of selection. Here I'm showing a H12 scan, and H12 measures haplotype sharing between samples. So if a certain allele is beneficial and is under positive selection, you would expect that that allele will increase in frequency in a population over time. But because there is some linkage between this allele and the neighboring parts of the genome, you actually see that a certain haplotype rises in frequency over time. And that you can measure with this H12 statistics that looks at an excess sharing of haplotypes. And that is an indication that at that locus there has been recent positive selection. So how do you read that? Well, we have this H12 statistic that we compute along the genome. That we compute along the genome, and we do that separately for each of the cohorts. And in these peaks, those are signals that there might have been recent selection over there. And within these peaks, we looked which genes we find in these peak regions. And in almost all cases, those are known as these peaks contain some genes that are known to be involved in insecticide resistance. And it seems also that indeed these genes are what's driving the selection. Okay, so now we do a little detour because we're interested in selection, and most of the selection will be for insecticide resistance. But we're also very interested when this insecticide resistance started to appear, because insecticides have been used since, I believe, the late 50s. And pretty soon after they started to be used at a large scale, there were reports of insecticides resistance in mosquitoes. So to address this question, we've also sequenced some historic samples. These were from museum collections at the London Natural History Museum and the IRD in Montpellier. And they are from the locations that you can see over here on the map. They are, we have like, you have what you have in that collection. So the numbers are not very big. We don't have like the big cohorts that we have for the modern samples. But we make do with what we have. And if you include these historic samples in a PCA, you can see that all of them kind of fall with where they would be expected to fall. So there has not been great change in this sort of very coarse background structure that we see. Okay, so let's look at some of the resistance loci that we see. So the first locus I want to discuss is the GSTE2 locus. And it has been known that there is one mutation, the L119F mutation, that increases the efficiency of metabolizing DDT and permethrin. So what we did here is we took these haplotypes that I discussed before. So those haplotypes are from statistical phasing. So rather than just knowing the genotype at each position in the locus, we actually know which variants belong together on the same haplotype. And here is a dendrogram where we did haplotype clustering within the peak region. And in this case, this contains 7 GSTE2 genes. And in this clustering, we expect that if there has been recent positive selection, we will have parts of this tree with haplotypes that are super similar. So for instance, like here, we see that all these individuals or all these haplotypes are basically the same, or at least very few differences. And if we then also look at which of these haplotypes actually carry this, the variant that is known to have an effect on insecticide resistance, we also see that over here, this cluster, where we have a lot of haplotypes that are exactly the same, most of them do have this variant. And the same over here, we have another small cluster that has this, this variant. A nice way of looking at haplotype trees like that is that you can very clearly see whether, if you see resistance in multiple places, whether that is from the same origin, in which case you would see only one cluster of very similar haplotypes, or whether there has been multiple origins and actually multiple haplotypes have been under positive selection. So here we see 2 clusters of haplotypes that have been under positive selection. And this resistance variant we find primarily in Western and Central Africa, but we also find it at some low frequency in East Africa. When we looked whether this variant was present in historic samples, we don't find any in the historic samples. So yeah, that's might be unlucky because we have just very few samples and we have not captured the resistance, or at this point, this resistant variant was not very widespread, and that's not, we don't know whether there has been another variant that was widespread at this point, or whether there was at this point just not so much resistance in this gene. Okay, then there's another gene, GABA, and this locus is also called RDL for resistance to dieldrin. GABA is a very big gene, so in this peak region, we looked which genes were present there, and there's one very long gene, the GABA gene, and then if we do haplotype clustering, we find a very clear cluster over here with haplotypes predominantly from Ghana, Nigeria, and some from Cameroon. And you can also see that those are indeed the locations where this variant, which again has been known from studies on the gambiae complex, that this variant deforms the receptor binding site and therefore can confer insecticide resistance, resistance to dieldrin. Okay, so if we now look at the historic samples, we do find that there is a couple of samples from the 1960s in West Africa, so sort of in the same region where you find this variant now at some considerable frequency, where this same variant is present in the historic samples. So that shows that this is quite an old variant that has been around since the late 1960s and is still around at quite high frequencies in some populations. Okay, then let's turn our attention to the next topic. One other thing we assessed in this data was whether the levels of variation that we see in this data set can be informative for gene-drive target identification. So for gene-drive targets, a part of the genome where it then will insert a drive and that will spread a gene drive allele through the population and then either will crash the population or will modify the population to be less able to transmit malaria. But what you really want for the gene drive target site, so the gene drive binding site, is that it's very conserved throughout the population, such that this target, the complement of this target, will fit in most of the individuals. If there is variation, then the target might not bind in some of the individuals, and then will make the gene drive a lot less efficient. So what we did is we looked for potential Cas9 target sites, and then we assessed whether there were any in this target site region, which I think is 20 base pairs long. We looked whether there is any variance in this 20 base pair sequence that then might undermine the efficiency of the gene drive. So we start, so here we check whether there is variation per population. And we keep adding in individuals from each of these populations. And as you add more individuals, you will see more variance, and you will be left with fewer targets that do not have any variance. So over here, for the different populations, we check at the number of Cas9 targets that you are left with if you take into account the genotypes of all the individuals that we have. And if you start with very few individuals, you get some 2 million potential Cas9 targets. And if you add more, you get fewer and fewer. And here is a zoomed out version of this number of Cas9 targets, which also contains in black all Anopheles funestus, and in gray, all Anopheles gambiae from the AG1000G phase 1. And what I want to draw your attention to is that the trajectory of the number of targets in and how that decays with increasing the number of individuals between gambiae and funestus is very similar. And the same for the number of genes that contain potential Cas9 targets. Also, the decay between funestus and gambiae is very similar. So I looked at one Cas9 target sites in particular, and this is in the double sex gene. So here, a population crash gene drive has been developed in Anopheles gambiae, and it's been tested in the lab in small cages and large cages, and it seems to be working very well. And I wanted to see what the sequence and what the levels of variation are in Anopheles funestus compared to Anopheles gambiae. So this gene drive targets the double sex gene, and in particular, it targets the sequence that is on the boundary between, I think between exon 5 and the intron before that, but don't pin me down on the numbers. And here on top, we see the sequence that is in the Anopheles gambiae reference genome. And below that, the sequence that we find in the Anopheles funestus reference genome. And they have one difference here in the intron and are the same otherwise. And then here we assessed whether, if we take into account all these mosquitoes from our data sets for funestus and for AG1000G phase 1 for Anopheles gambiae, what kind of variance we find. So for funestus, we find only one variant at very low frequency, 0.8 to 1.5% allele frequency in 2 different cohorts. And actually, this variant is the reference, the one difference between the reference sequences in funestus and gambiae. But in gambiae and Calitzi, we find 2 variants. One might be a sequencing error because it's only found in one haplotype in one individual. But the other one seems to be very real, and it's found in 4 different cohorts, and in Calitzi from Angola, actually at quite high frequency. That does not necessarily mean that a gene drive will not work in that population, but that is really something to take into account and assess what the effect is of variation in the target sites and whether there is any way to remedy that will lead to decreased efficiency of the gene drive. But overall, the amount of variation in this gene drive target in Anopheles funestus is extremely low, and that is a good start to see whether the same approach can be adapted to also be used in, as the same gene drive approach can be adapted to also be used in Anopheles funestus. All right, so with that, this has just been hopefully the start of what is going to be very regular genomic surveillance of Anopheles funestus. And with this work, we hope to sort of have created a baseline understanding of the structure and variation and insecticide resistance status in Anopheles funestus that can then be used for further genomic surveillance work. The things that we hope to really improve is that with improved spatial sampling, we can sort of see what Anopheles funestus looks like throughout all of its range, and try to understand why sometimes at small scale, like South Benin and North Ghana, certain populations can suddenly look very, very different from their neighbors. Also, we want to have to increase the temporal sampling scheme to see how this population structure might be changing over the years and throughout the seasons, and whether some of these odd populations are persisting through time. And then in the future, I think it's also going to be really important to integrate phenotypic information to have some understanding of whether any of the unexpected structure we see might be related to adaptation, and particularly also for insecticide resistance, it's going to be very important to have resistant phenotypes to accompany the genomic information. And then to be able to have a better understanding and higher resolution of the genetic structure we see, and in particular the phenomena that we right now can't explain very well with just SNPs. It's going to be pivotal to have long reads for some representatives of these populations. And I think with all of that together, that's going to have a very positive effect on genomic surveillance for all of the important malaria vectors. And with that, I would like to thank a lot of people. As I said, this paper has really been a collaboration between a lot of people, and a lot of people put a lot of work in it. So here on the right are all the co-authors on this paper. And it's been great to work with all of them. And then in particular, I want to thank the people from Mara's team, so Alex, Mara, Joachim, and Petra, who have worked a lot to make this paper possible. And I also want to thank Petra for all the wonderful drawings that you've seen throughout this presentation. And with that, thank you for listening, and I'm very happy to take any questions.
Thank you so much, Marilou. This was brilliant and fascinating. Thank you so much. We've now reached Q&A as well as discussion time. So kindly either raise your hand and we'll give you the right to speak, or type your question in the Q&A or chat box, and Marilou will address your question. So we have about 10 minutes for Q&A, as I said. Okay, I see Meza has a question, and I'll ask a question after. Meza, go ahead.
Hello, everyone. I'm audible?
Yes.
Okay, thank you very much. It's a very great to see your work, a huge work on malaria. And basically, I'm from Pakistan, an Asian country. As my concern is that, as you are working in sub-Saharan Africa, is according to my knowledge, if I'm not wrong, it is one of the highest titer over there of malaria. The second highest titer of malaria over here in Asian countries, especially in India, Pakistan, Myanmar, and some other countries have the highest ratio of malaria. So my question basically is that we are working over here on malaria, and last year we have developed some techniques to detect the malaria with a actually we are working on its diagnostic and therapeutics as well. So we are working on diagnostic and therapeutics as well. What we need, we also request to Sangha Institute to develop a surveillance center here in Asia. So my concern is that if we move forward to develop a sequence center or a surveillance center here in Pakistan for the Asian countries, what we get, how we get connected with the Sanger Sequence Institute, and how you can help us. Is there any possibility from your side?
I think it would be great to have also genomic surveillance and to try and develop more genomic resources for malaria vectors outside of sub-Saharan Africa. I would encourage you to get in touch with the people at Sanger that they might be able to advise you. I'm not really involved in policy decisions, but it's definitely good to reach out. And even as you, another thing, like one thing is the sequencing, but the other thing is to also be able to do all the analysis, and actually in that case reach out to Pabalo, because the Malaria Gen and PAMCA together are running a fantastic training course to analyze genomic data, and you can be involved in the training course, but also all the trainings are available online on the Malaria Gen. You can get there via the Malaria Gen website. But yeah, regarding sequencing and advice on how to set that up and how to get budget for that, I would reach out to the Genomic Surveillance Unit and ask them.
If you don't mind, can I get your email address so I can in touch with you and also get some advices from you as well?
Meza, just to jump in, I think there's a comment from Petra in the chat saying you should email ANOSP. So there's a link there that you can click on. And I would also advise you to email info@sanger.ac.uk. So click on the link that Petra put in the chat and kindly email info@sanger.ac.uk. They might be able to advise on that.
Oh, thank you very much. Thank you very much for your time, please.
Thank you.
Thank you.
Thanks, Petra. Petra has also put the email in the chat. Thank you so much, Marilou, for that answer, and thank you, Mesa, for your question. We still have about 7 minutes to go for Q&A as well as discussion. And there was a comment in the chat saying, thank you so much for your presentation, which was clear. That's from Sedeche Lebrage. That's for you, Marilou. And I have a question. I'm also trying to see, there's a comment from Todd. Thank you very much, Doctor, for your interesting webinar. It has been very inspiring. So just to echo that as well.
Thank
Marilou.
you very much.
My question is, I think I saw that in your study, you used modern samples, about 600, and I think 45 samples that are historic specimens from about, some of them 98 years ago, if I'm not mistaken. So I just wanted to know, were there any particular challenges or difficulties with sequencing historic specimens, especially knowing that sometimes or most times with historic specimens, they tend to have fragmented or damaged DNA? Were there any of such challenges in your sort of study, and what was the impact on your findings and your study in totality as well?
Yeah, working with historic DNA is very challenging, and really Petra deserves all the credit for doing that, from the extractions to the data analysis. It's hard to get very good coverage in sequencing these samples, and some of them have like only, so for the modern samples in this study, we have between 30 and 40x coverage, meaning that on average each base in the genome has been covered by 30 to 40 reads. With the historic DNA, some of those have only 5x coverage on average. So it's much harder to reliably call variants. And you also can't just include these data in the analysis that you're doing. So for instance, the PCA I showed you, if you do that in the same way that you would do for the modern PCA, then the main split you're going to see is between historic and modern samples, because of these artifacts caused by sequencing historic DNA. So what we did instead is that we used the modern samples to sort of have a background understanding in this PCA space that sort of is indicative of the structure that is important in all of these things. And then we projected the ancient samples on top of that. And I think that's sort of been a recurring story, that you have to be a bit creative in how you do your analysis to not let coverage, higher error rate, and other factors like that drive the results that you see. So for instance, for the insecticide resistance studies that we did, we couldn't include these samples in the haplotype trees because we couldn't phase haplotypes because they have so much missing data. So what instead we did was that we used the modern samples to identify some variants that seem to be under selection and seem to be important. And then we went back to the genotype calls and sometimes even to the mapped reads in order to see whether we see that variant in ancient DNA and whether we think it's real and not an error or an artifact, because ancient DNA also degrades and that can also introduce things that look like variants, but really that only happened after the mosquito died. So that's not informative for variation in a population at that time. So yeah, sometimes we had to go back to the reads and see whether the variants we found, the variant calls that we found for the historic samples seem to be genuine.
Okay, thank you so much, Marilou, for that comprehensive answer. There is a question from Dr. Simpson in the Q&A. She's asking, what do you think the next malaria Anopheles species of focus should be?
I don't know. I think, well, so first of all, I am much more familiar with vectors in sub-Saharan Africa than vectors in other regions. And as Mirza just said, I think there is a great potential to also study these vectors from other places. But I think what's also very important is I think it's very important that we now have to work for those vectors that we know are very important in transmitting malaria, to have a good understanding per species of how the structure is, what resistance there is, etc., what the levels of variation are. But I think there's also great potential in looking at all the other species that seem to have some wide species range, and that there's a hint that they might be involved in transmitting malaria. And rather than sequencing thousands of samples, to sequence tens of samples and have a little bit of an understanding of all the secondary vectors that are out there, yeah, what might be going on in those.
Okay, thank you so much, Marilou. There's a thumbs up from Dr. Simpson. I'm trying to see if there's a lot of, okay, one last question because I'm looking at the time, so I'll just allow it. There's a question from Kiprono Korir. I'm Kiprono Korir from Nairobi, Kenya, currently doing my master's. Malaria causes havoc in sub-Saharan Africa, and I've always thought of vector control strategies, including understanding their backgrounds. Could there be a chance to work in your lab? That's a question from Kiprono.
Well, I myself don't have a lab at the moment, so that's going to be challenging. But I do think that it's very important to collaborate between people from different countries with different areas of expertise and different backgrounds. And there is a lot that we can learn from each other. And that really goes both ways with analysis. Like, I might have more experience in doing analysis, but I know a lot less about these vectors and how they sort of where to find them, how they change over time and through the season. So I'm very keen to collaborate, and I think again I would like to mention the genomic training courses that are organized by Malaria Gen, because I think that's a very good way to get into doing genomics and learn about that, but also a really good way to meet other people who are interested in working on the same system, and sometimes from very different backgrounds. But yeah, you get to meet loads of people from all over the world, and it's a very good ground for setting up collaborations also.
Okay, I'm just going to squeeze in one last one because I'm aware of the time. There's a question from Gregoire Sawadogo, and they say, thank you for this great presentation. Could you please explain more on the screening of gene drive targets? Thank you. That's from Gregoire.
Well, so very briefly, we looked for sequences of 20 base pairs that are in the coding regions of genes that have no variation in all of the individuals we see, and that answer with the PAM, what does it stand for? The proto-adjacent motif. I think it has to end in, okay, I'm not going to say, in two nucleotides that are the same. So it's a very crude way of doing that because we don't take into account what the functions of the genes are and how easy or hard it might be to develop something that binds exactly there. But if you want to know more about it and more in detail, then please check out the preprint, and there all the methods are described in more detail also. Thanks.
Okay. So I've shared the link to the training course that Marilou referred to. Please check in the comments. And I also shared the link to the preprint. Please do go through that. And yes, Marilou is keen to receive your emails. Please follow her on Twitter as well. But do email her if you have any further questions. And if you want to know about collaboration and any sort of explanation as well in her work. Thank you so much, Dr. Marilou Boddé, for your fantastic, engaging and informative presentation. Do you have any last words before I close off?
Well, thank you very much for inviting me to give this talk, and I'm always very happy to talk about mosquitoes, and it's been great, and thank you for all the questions, and thank you for your very kind words, Pavello.
Thank you so much, Marilou, and thank you so much to all of you attendees for attending the first General Club for 2025. See you next month on the 18th of February for the second one, for the second installment of the year, where we're going to have Sophia Minyi from Tanzania presenting on the new cryptic taxon or mosquito that is found in East Africa. So please be on the lookout on our Malaria Gen, GSU, LinkedIn and Twitter pages, as well as our websites for details on that and on how to register. But yeah, otherwise, thank you so much, everyone, for joining. Enjoy your day and see you next month. Thank you so much, everyone.
Thank you, Pavello.